two more topics left in this chapter: grams of active fast stain and special stains. And that's it. Then we start chapter 4. We have this week, we have next week, and then let's set up the date for exam 1. Okay. Okay, who has the calendar? Not next Tuesday, but the following Tuesday. What's the date? The 29th? Not next Tuesday, but the following Tuesday. The following Tuesday. February 5th. Tuesday. Fifth. Okay. And what's the Saturday before that? Thank you. Most likely in this classroom, okay, but I will have to double check if this room is available. So most likely right here, 12 to 2, or maybe 3, 12 to 2 to 3, yeah. So probably most likely 3 hours. It will go by, I think it will last for maybe 3 hours, yeah. Yeah. Okay, any questions?
When you are going to send us the link for lecture?
Or the new, yeah, pretest? Yeah.
Or the link for a lecture? YouTube video.
Oh, they're already two. Yeah, I know. As soon as I get them, yeah, as soon as I get them, I send it to you right away. I have not received the other links. Yeah, so I think because of the holiday, that's why. So I hope that I get the other 2 today. Okay. Yeah. And I will send you the pretest. One of you should just send me an email. One person should send me an email for the pretest for exam 1, and I'll send you the new pretest. Okay? All right. Questions? Gram stain is a differential stain. True or false? True. Gram stain is a differential stain. True. It differentiates bacteria into 2 groups, gram-positive and negative. Gram-positive bacteria, they have positive charge, and gram-negative bacteria, they have a negative charge. Good. Good. All bacteria, they have a negative charge. Good. Okay. Gram-negative bacteria have more lipid in their cell wall.
True.
Negative. True. Gram-negative bacteria, they have more lipid in their cell wall. That's why when you decompose with alcohol, they lose their lipid, and that's why they appear pink. Okay. Gram-positive bacteria, they have more peptidoglycan in their cell wall. True. Okay. So gram stain differentiates bacteria on the basis of lipid and peptidoglycan in the cell membrane. Cell wall. Not the membrane, cell wall. Yeah. Cell wall, not the membrane. Okay. Two factors that may cause, yeah, I know that, yeah. Two factors that may cause gram-positive bacteria to appear gram-negative. Very good. Over decolorization, old culture. Good. Some of the slides that appear, again, gram-negative slides this morning, they turned out purple. What was wrong with those slides? Okay, here's the answer. Gram-negative slide that turned out purple, they were gram-variable. True? Now remember, gram-variable is a term only applicable to gram-positive bacteria. If a gram-negative bacterium is purple, it is under-decolorized. Under. Remember, they are supposed to lose their purple color. If they did not lose their purple color, you'd under-decolorize. You did not apply alcohol long enough. Long enough. Long enough. Can you fix it? Can you fix that slide? You have already applied oil on it. Can you fix it? We actually did this morning, yeah. Actually, that part on which the oil is, that's ruined. But your smear is pretty good enough. Okay. The rest of the smear can be fixed. So this morning we did that. They're looking at a slide. So I told the student, remove the slide, just dab the oil, and go ahead, decolorize it for another 15 seconds, put safranin on, and your slide is fixed. And that's what we did. Cool. Yeah. You could do that. Yeah. Instead of starting over, making a smear, doing the whole gram stain, fix the smear, observe it. We did it.
I understand what you just said about a gram-negative.
I have no clue what I said.
Gram-negative can be under-decolorized because you haven't put enough alcohol. How do you know that it's not a gram-positive?
Standard time is 15 seconds. If you have applied 15 seconds, you don't have to worry about it. After 15 seconds, if your bacteria is still holding on to purple color, you have no doubt. But if you did not apply alcohol for 15 seconds, only for 5 seconds or 10 seconds maybe, okay.
Why would you, I mean...
You see, I have already told my class that, okay, which bacterium was which. If you did not know, that's a different story.
That's what I'm saying. Most of the time you don't know.
You don't?
All the time.
But experienced microbiologists, they will know. Like, for example, this morning when I did the slide in front of you, right? How long did I apply? I did not even time any of the reagents. Did I time? No. Okay, so, yeah. So, all right. Or any other questions? All right. So let's finish up chapter 3, acid fast stain. Before I show you the acid fast stain, let me tell you when would you do an acid fast stain. Acid fast stain is for special bacteria. Question? Go ahead. No? Okay. All right. Acid fast stain is for special bacteria that cannot be stained well with gram stain. Yeah. There's a reason why not all bacteria can be stained with gram stain. Okay. Some bacteria, they are called acid fast. What does that mean? Acid fast. Acid resistant. They are so tough that their primary color cannot be removed with acid. They are so tough. Okay? In gram stain, alcohol cannot remove the primary color, But these bacteria are so tough that even a mixture of acid and alcohol cannot remove the primary color from their cell wall. They're so tough. That's why they're called acid-fast, acid-resistant. Acid-resistant bacteria. And there are only two types. Number one, mycobacteria. And number two, nocardia. What is so special about these two bacteria? Okay. These two bacteria, they have this wax-like chemical called mycolic acid. Mycolic acid in their wax-like, waxy lipid. Waxy lipid in cell wall. Because of this, they cannot be with gram stain. It's like if you take a wax candle and stick it in crystal violet or saffron. Would you have a homogeneous layer outside it? No. It will be beaded on the candle, right? That if you try to stain a bacteria that has mycolic acid, it will not stain well. So these bacteria, acid-fast bacteria, sometimes they are also referred to as poorly gram-positive. Why poorly gram-positive? Because they do accept purple color, but it's patchy. Poorly gram-positive bacteria. How do you identify them? That's one characteristic, that they are poorly gram-positive. Second characteristic of these bacteria, acid-fast bacteria, is that they are, they are pleomorphic. Pleomorphic. Pleo means many. Morph means shape. Most bacteria that we encounter every single day, E. coli, staph, strep, if you look at their pure culture, like you looked at staph today, right? If you look at the pure culture, okay, all bacteria in this culture, they are round shape, right? This bacteria is called monomorphic. Most bacteria are monomorphic. They have single shape in one culture. Monomorphic. One morphic shape. One shape. But if you look at the culture of acid-fast bacteria that cause TB, okay, leprosy, this is what their shape is. Look at this. This is pure culture. This is not lint or any other thing. It's not fungus. This is pure culture of acid-fast bacteria. Pleomorphic. Many shapes of same bacteria. Pleomorphic. So these two characteristics, let's say if you're doing a gram stain, okay, gram stain, you see two characteristics of your stain. Let me erase some of this. You take the sputum of a person who has TB. You make a smear and you stain it. Okay? You will have, do I have purple? I don't have purple color. So you'll have nice monomorphic bacteria that are going to be purple. Okay. You may have some rod-shaped bacteria that are also monomorphic, purple. And then on this slide you will also see some weird shape. Not only that, patchy, not homogeneous purple color, patchy. Because of mycolic acid, the purple primary color is not going to penetrate into the cell wall. So these two characteristics, poorly gram-positive and pleomorphism, this tells you that go ahead, do what? Acid-fast stain. Now let me show you what is acid-fast stain. We'll come back and we'll look at the principle of that. What is acid-fast stain? Do I have that on there? I don't think so. Let's just do it right here. Acid-fast stain. Now, from last time, let me ask you, in differential stain, how many stains do you use? Two. Okay. So we are going to use two stains of contrasting color, just like gram stain. But unlike gram stain, acid-fast stain uses only 3 reagents. Instead of 4, we use only 3 reagents. Acid, fast stain. All right. You will do this stain in lab next time. First, you make your smear, okay, sputum smear, okay. Air dry, heat fix. After heat fixation, you place your smear on steam bath. Okay, hot plate, or you can just make your homemade steam bath. Coffee can, or tray on the coffee can. Put your slide on the steam bath. Now you pour your primary stain while your slide is sitting on the steam bath. What's the purpose of steam bath? So let me just put, say. Exactly. Place slide on steam bath. To loosen up wax, to allow the primary stain to penetrate into the cell wall. That's the purpose of steam bath. Loosen up, okay, cell wall, which is made up of wax, waxy substance. Then you add your primary stain, which is carbol fusion. Add carbol fusion. Carbol. That's our primary stain.
I'm sorry, the stain that pushes out the cell membrane or the cell wall?
Cell wall. Cell wall. Yeah. Target of all stains in bacteria is cell wall. Okay. Okay. And you let it sit for about 3 minutes. Let it steam for about 3 minutes. Make sure that you stay with your slide. Don't let it evaporate. If the stain dries up on your stain, you start over. It's a very nasty stain. Keep your slide moist. If the stain is evaporating, you add more stain. Keep it moist all the time.
What do you? I'm not confused on the steam bath. What are you using for steam bath?
So is this is what we use. This is our hot plate that we use in chemistry. Coffee can. In the coffee can we have water, and here is the staining rack, and here's your slide on the rack.
Got it.
Okay, all right. That's my beautiful diagram. All right. So after 3 minutes, let me ask you this before. That's more important. All right. Color of acid-fast bacteria, non-acid-fast bacteria. Pay attention to the last two columns. Fair question on the exams and the quiz. Okay, make sure you understand the last two columns. Okay, positive bacteria. This is acid-fast. This is gram-positive and gram-negative. Okay. First of all, before I continue with this, what is non-acid-fast bacteria? How do you define non-acid-fast bacteria? What is it? Any bacterium that does not have mycolic acid in its cell wall. Any bacterium that lacks mycolic acid in its cell wall is called non-acid fast. Okay? How about gram-positive fast? Yes? They don't have mycolic acid. Gram-negative, non-acid fast? Absolutely. So both gram-positive and gram-negative, they are non-acid fast. Non-acid fast. So why did I put them in separate categories? Explain that.
On the basis of their lipid and...
Very good. Because they have different lipid concentration. Okay, and I will ask you a question later that will explain that. So, after 3 minutes, some books, they may say 5 minutes, but 3 minutes are good enough. You wash it with water. So what is the color? What is the color of acid-fast bacteria after the primary stain? Oh, by the way, carbonyl fusion is magenta color. Beautiful purplish red color, magenta. What's the color of acid-fast after primary stain? Non-acid fast? Remember, how many stains have we put so far?
One.
Only one. So everything is magenta. And remember my statement last time that first step of all differential stains is so acid-fast stain is also a differential stain. First step is simple stain. So everything is magenta. Magenta. Magenta, magenta. Make sense? We have only put one stain on, no other color. All right. Here comes the most crucial step of this staining procedure, which is decolorization. And for decolorization, we use a much stronger decolorizer. In gram stain, what do we use? 95% ethyl alcohol. For acid-fast stain, we use a much stronger decolorizer, which is called acid alcohol. Decolorize with acid alcohol. How long? 5 seconds. Some books may say 15, 30 seconds. My experience, 5 seconds. 5 seconds or less. 5 seconds. Or less. Because if your smear is really, really thin, you may just have to add it for maybe 2 seconds. Add it to your smear, wash it away right away. 5 seconds or less. By the way, what is that? 5% hydrochloric acid, 5% HCl, and 95% ethyl alcohol. 95% ethyl alcohol. Okay, that's your acid alcohol. All right. Now please tell me the color of acid-fast bacteria, and then after about 5 seconds you wash it right away with water, right away immediately. Wash with water. Okay. So what is the color of acid-fast bacteria? Remember, acid can't remove the primary color. Acid resistant, so it will remain magenta. It is so tough that even the acid and alcohol cannot remove it. So magenta. Magenta. How about the rest? Non-acid fast. They have no mycolic acid. Colorless. Colorless. Clear. Colorless. Clear. 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 Same principle like gram stain now. To make these bacteria, clear bacteria, more visible, the counter stain in acid-fast stain is methylene blue. Methylene blue. Okay? Now you add methylene blue.
That's the third reagent, right?
Just right. Methylene blue. 1 minute. Wash after the methylene blue. You wash, you blot, and you observe your slide. Now when you mix magenta, purplish red color with light blue color, which one is more dominant? Magenta color. So magenta bacteria remains magenta. And clear becomes blue. Blue. Okay. Now let me show you the picture. If you have the atlas handy, may I borrow your atlas? You have atlas? No atlas. Sandy atlas? Okay, got it. On page number 7 in your atlas, please. Page 7. Acid fast stain. Ren, if you could please turn these lights off for me, please. Thank you. Yeah, that's good. Okay. This is acid-fast stain. You can see, first of all, magenta color, acid-fast bacteria. Notice the pleomorphism.
Many shapes.
Man, look at this one. Look at this. Look at this. There's nothing wrong with these bacteria. But this is called pleomorphism. Look at this. Look at this. Magenta color. And here's your non-acid fast bacteria. Blue color. That is acid fast stain. Make sense? Okay.
The rods are the acid fast.
Pleomorphic rods. Pleomorphic rods right here. All right. Let's see what you have in your textbook. Is there a picture of acid fast in your textbook? Nope. Oh, they do, but doesn't show pleomorphism very good. Right here, there is a picture. It does show two colors. I don't know if it shows pleomorphism that good. Maybe, I don't know. I like mine better. Okay. It does show, because of pleomorphism, unless you break down the big chunks of mycolic acid, this is how your smear is going to look like when you do it next time. So you need to de-chunk your slide. Really, new word, de-chunk. You need to take your loop, break down the wax into little pieces. Okay, so you can see the individual rods, or this is mycolic acid holding this rods together. You see this ear wax is because of mycolic acid. There's a bacterium in your ears that lives in your ears and external genitals called Mycobacterium smegmatis, from smegma, that produce the smegma, sticky stuff. Okay, and Mycobacterium smegmatis in your ear wax. Aliens don't come and put wax in our ears. No. Mycobacterium smegmatis. Okay, so that's acid fast stain. Now the special stains. Last topic. Oh, before I talk about last topic, I'm going to make one change here and then ask you this question. Decolorizing bottles, both acid fast decolorizing bottle and gram stain decolorizing bottle, ethyl alcohol bottle and ethyl alcohol bottle, they look identical. The labels are different. Okay. The labels are different. Let's say your lab partner is a prankster. You are doing acid fast stain. Acid fast stain. But your lab partner switches acid alcohol with this. Tell me the colors now, please. Only this step is different. Instead of using acid alcohol, you are using what? Ethyl alcohol. Tell me the colors of these bacteria, please. I'm going to erase, no, not erase, but write the colors in green. So you are using this. So now give me the color of acid fast bacteria.
Magenta.
Magenta, very good. Magenta. Magenta. Now this one, non-acid fast, I'll skip, because this time you have to divide them into gram-positive and negative category, because that's where you will see the difference. Okay. Gram-positive will be what?
Magenta.
Excellent. Magenta. And gram-negative will be? Clear. Excellent. And this is why we use acid alcohol for acid fast stain. We do not use ethyl alcohol because these two bacteria, okay, they have the same color. If you use acid, you must use acid alcohol. Yes, yeah, mixture. Yeah. Yeah. Okay.
So what would the bottom row be?
Bottom row will be, okay, these two will be magenta and this will be gram-negative, will be blue. Gram-negative will be blue. Yeah. This will be magenta, magenta, and this will be blue. Yeah. If you do not use acid alcohol, if you use ethyl alcohol instead of, that's a question in your lab handout. this is the question. What if you do not use ethyl alcohol if you use ethyl alcohol? Okay? Why can't you use ethyl alcohol? That's the question in your lab handout. All right. Any question on fast stain? Okay. If someone asks you, name one acid-fast bacteria. I know Mycobacterium tuberculosis is a good example. Mycobacterium bovis is another. But you know Avis carvental? Avis? There's a bacterium that can cause a TB in birds. Avis mean bird. If you have, birds, Avis, Mycobacterium avis is a bacterium that can cause TB in birds. So remember, that's very easy. Mycobacterium avis. Okay? Or Mycobacterium leprae from leprosy. Leprae. Okay? All right. So that's the end of acid-fast stain. Now, special stains. Question. When you do a simple stain, okay, let me put my notes up so you can see. When you do a simple stain or a gram stain or an acid-fast stain, why can't you see all different parts of a bacteria? For example, bacteria have flagella, they have fimbri, sometimes they also have a gelatinous layer called capsule. Okay, we are done with that, we are done with that. This is the one we are talking about. Here we are. Okay. Last point right here. This is the one we are talking about. Special stains. Okay. Question to you is, when you do a simple stain, gram stain, or acid-fast stain, okay, and you want to see, let me show you what we are talking about. You want to see this right here. There's a bacterium. You just did a gram stain, right? And you wanted to see this pilus, this hose-like structure right here. Okay? Or you wanted to see this whip-like structure right here. When you do a gram stain or acid-fast stain, why couldn't you see these structures? Very good. The target of the gram stain is what? Right here. The gram stain that sticks to the cell wall. Every stain has its own target because, chemically speaking, every part of this bacterium is made up of different chemical. Chemical composition of each part of the bacterium is different. If you want to stain flagella, sure, you can observe flagella under bright field microscope, but you have to use a stain that has chemical affinity for flagella. Crystal violet doesn't bind to flagella. There's a special stain that binds to flagella, sure. If you want to observe capsule, capsule, this gelatinous layer, the outermost layer, capsule. How do you stain capsule? That's a little tricky, okay? We'll talk about that in lab, but by the way, just no stain sticks to the capsule. Can you think of any reason why no stain will stick to this capsule? Because they don't have any charge. Capsules are non-ionic, so you can never stain capsule. You can never stain capsule. Okay? So what's the best way to observe capsule? Okay. Okay. Here we go. Negative stain. And next one. You can see capsule under negative stain, sure. Here we go. Capsule. Stain the background. Beautiful contrast. Stain the bacteria, stain the background. Beautiful, excellent contrast. Okay? So it's a combination of two stains actually. You stain the bacteria and you stain the background. Okay. But you cannot never stain the capsule because they have no charge. It's just like clear halo. Clear halo. Okay? So the question was, why can't you observe all the other parts of the bacteria when you do gram stain, simple stain? Because each part of the bacterium is chemically different. And different dyes, they bind to different parts of the bacteria. If you want to observe flagella, you have to use a dye that stick to the flagella. You want to observe capsule, you don't have to do the dye. You have to use a special technique to observe capsule. But if you want to observe other parts, you have to, like endospore. When you do, okay, here's a tricky question, okay? Quiz or on the test. Can you, listen to the language. Don't write it. Okay? First listen and then you can write it. Can you observe endospore by doing simple stain? Okay. Can you observe simple stain by doing simple stain?
No.
Okay. Can you observe? Okay. Endospore by doing simple stain? The answer is yes. Okay. What will be the color if you are using crystal violet? You're doing crystal violet to do a simple stain. Okay. Very good. David said it will be clear. Because crystal violet does not bind to endospore. Bacteria will be, let me actually show you. Some of you, I did actually show you. Oh, here we go. Here's the bacterium that makes Botox. Botox. Okay. Really? Yeah. Look at this. This is the bacterium that was stained with crystal violet. Okay. Here's the endospore. What's the color?
Clear.
Clear. What's the color of the rod? Purple. So yes, you can see the endospore by doing simple stain. Okay? But if you want to see, if you want to stain the endospore, here's the endospore stain right here. What's the color of the endospore? Green or bluish color, aqua color, right here. Okay? So yes, you can see endospore by doing simple stain, but it's not stained because the stain is not for the endospore. Here, there's a special stain called green that sticks to the chemical in the endospore. Okay? So special stain. The purpose of special stain is what? To observe special part of the bacteria. Those are special stains. The end. Okay? Yes?
How do you say this is endospore? Can you say this is flagella if it's not correct?
Oh, you have to at least know the location of these parts, right? Yeah. You have to know the location, right?
The question that you asked is, can you absorb endospore by simple stain?
I never said that thing at all. I never used that word, absorb. Yes, thank you. Right, yeah. Go ahead, say, I'm just picking on you. Yeah. Go ahead.
And your answer was
Yeah.
yes with crystal violet, but the yes means you can only see like a, it shows up clear.
Yes. Yeah. Yeah. Right. Remind me to show you that. No, actually, I will show you when we do the endospore. I'll show you both slides, endospore stain and simple stain for comparison.
But
Okay.
any other coloring, they won't show up other than what it's made for.
Correct.
You won't
Correct.
see anything.
I'm not sure if I follow your question. Go ahead, repeat it. No problem.
Use another color other than crystal violet.
Yeah. Like safranin. If you use safranin for endospore, what is the color of endospore? Safranin is red. What is the color of endospore? Clear. Because safranin does not bind to the endospore. It binds to the bacteria. You must use a stain that binds to the endospore. And there's only one dye that I'm aware of that binds to the endospore called malachite green. If you use that, it's going to be green.
Okay, but I'm not understanding.
Me neither.
The question you asked was, can you observe endospore by simple stain,
Answer is yes.
but only with crystal violet?
No.
Oh, that's
No.
what I...
No. You can use any cationic stain. any cationic stain to observe endospore. But the endospore will be clear, colorless. Endospore will be colorless.
So if it's colorless, how are you supposed to see it?
Very carefully. I will show you, okay? When you see it, okay, you will have better understanding. Yeah, sometimes it is hard to explain, but I will show it to you, okay? All right. Chapter number 4. This is where most of your exam will come from, half of your exam. Okay, chapter 4. First half is memorization, which I really don't like, but there are certain things in microbiology where you have to memorize, like the size, shape, and arrangement. Size, shape, and arrangement. Foundation fundamentals that you have to memorize. So I'm going to go over that with you. Second half is understanding, okay, that you'll enjoy more. But let's go over the memorization part, okay? Difference between prokaryotes and eukaryotes. Holy mackerel. We are supposed to come back one at a time. No. Okay. Let me go back. Okay, fine. No problem. We will start with differences between prokaryotes and eukaryotes. You already know one difference from chapter number 1, right? And that is what? Prokaryotes, they do not have nucleus. Eukaryotes, they do have nucleus. And here are a whole lot more differences. Please make sure you understand these. They will definitely show up on your exam in a scenario format, okay, which I'm going to practice with you, okay, and of course memorization too. All prokaryotes, they have one circular chromosome, okay, but which is not enclosed in a membrane. So you can simply say that prokaryotes do not have a nucleus, okay. Are your notes different than this?
Yes.
Slightly. These are my new notes, okay. All right. Lot different? You don't even have those? So I'll go slow, you can write them.
So the first one is the same thing as saying
No nucleus. Yep, same thing. So I'll go slow, you can write them, okay? Okay. In eukaryotes, chromosomes are paired, and they are enclosed in a nucleus. How many pairs in humans? 23 pairs. In somatic cells, 23 pairs. Do you know the length of DNA in one human cell? Just the average length. If you pull one cheek cell or one somatic cell from your body, what is the length of that DNA? You can dangle it from your head to your toes. That's the length of DNA in one cell. And if you take the DNA out from the whole human body, you can moon and back. That's the length of DNA in our body. No histone proteins. Histones are types of proteins, but in prokaryotes, there are no histones. We do have histone. Anyone knows the significance of this protein? What is a histone protein? Okay, let's look at that. Okay, it has something to do with the amount of DNA. Histone proteins are triangular proteins, like this. Triangular proteins inside the nucleus, and they allow the DNA to wrap around it. DNA literally wraps around these histone proteins. In eukaryotic cells, this is DNA, and this is histone protein. Because eukaryotes, they have large amounts of DNA, right? 23 pairs. So our DNA requires organization. So the job of histone protein is to allow DNA organize itself inside the nucleus. Histone protein, it allows the DNA to organize itself inside the nucleus. Organization. And how many chromosomes do they have? Only one. No histone protein required. No histone protein. No organelles. Prokaryotes, no Golgi body, no endoplasmic reticulum, no organelles. Eukaryotes have organelles. Cell wall. Prokaryote cell wall is complex. Add the word complex. I think in your notes you do have the word complex. Mostly made up of peptidoglycan. Peptidoglycan. Make a note and remember it. Highlight it. Rigidity of the cell wall. Firmness, rigidity of the cell wall is because of this chemical. Peptidoglycan. You take this out, bacteria will lose its shape. Rigidity, firmness of the bacteria is because of peptidoglycan. If the bacteria loses peptidoglycan, it will lose its shape. Eukaryote cell wall, okay, those cells that do have cell wall, their cell wall is simpler, made up of polysaccharides, complex sugars, such as, have you stepped on a cockroach? You hear that crunchy sound? It's because of chitin. Chitin. Fungi, they have cell wall. Their cell wall is made up of mannan, okay, glucans, also chitin sometime, glucan, mannan, glucan, mannan, okay. These are all complex sugar. Okay. Plants have cell wall, right? Plant cell wall is made up of what? Cellulose. Cellulose. But these are all simpler materials as compared to peptidoglycan. And we'll look at peptidoglycan. Okay. Much more simpler. Cellulose, if you look at the structure of cellulose. What is cellulose? Long chain of glucose, 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 glucose with few side chains. That is cellulose. Metamucil. That is cellulose. Yep. Complex sugars. Why can't you get energy from that glucose? It's a polysaccharide, but we can get energy from starch. Same thing, polysaccharide. No enzyme. No enzyme. We cannot make cellulase like cows. Cows are constantly eating. Yeah. Secondly, cellulose has very little nutritional values. Have you ever seen a cow just not moving its mouth? Never. It's because they're constantly eating, because cellulose has very little nutritional value. Okay, that's why they're constantly eating. Okay. Anyways, all right. Reproduction. May I have it? I'm just going to step on it.
I don't know how do you turn off this?
I know, I'll just step on it. Okay. Binary fission. Reproduction. Okay. Reproduction is by binary fission. That is asexual reproduction. No sex, no gametes. All prokaryotes, they reproduce by binary fission. 1 cell to 2, 2 to 4, 4 to 8. That's called binary fission or asexual reproduction. No gametes are involved. Okay. Eukaryotes, true sexual reproduction. Is there asexual reproduction in eukaryotes? There are some eukaryotes reproduced by asexual reproduction. Fungi, some fungi, like yeast, they reproduce by asexual reproduction. Yeah. What is the disadvantage of binary fission? Disadvantage. Asexual reproduction. Very good. No variety. Grandma looks just like granddaughter, okay? No variation, genetic variation. Except for mutation, that's a different thing, okay? And what is the disadvantage of sexual reproduction other than stinky partner? DNA abnormality, loss of sources. How many sperms are released to fertilize one ovum? Millions. Okay. So loss of resources. Or you can think of others later. Okay. All right. Ribosomes will show up on your exam for sure. Remember, prokaryotes, bacteria, their ribosomes are smaller, 70S. S is a unit that measures sedimentation in liquids, called Sieved-Berg unit. Sieved-Berg unit. Don't have to memorize that for the test. Okay, I'm not going to ask you the unit. Just want you to remember that in prokaryotes, ribosomes are small. In eukaryotes, ribosomes are large. But, yep.
On that basis of volume of measurement is sediment, you said?
Sediment, yeah, sediment in liquid. For example, okay, now let me show you. I think if they have a picture, fine. Otherwise, I'll draw my own. Exactly, yeah. Oh, here it is. Yeah, they do have a nice picture. So let me show you this. And you answer this one if you could, please. Let's see. Already open. Fine. When ribosomes are not, what's the function of ribosome? They assemble protein, they make proteins, okay? Factory, their proteins are assembled. And when ribosomes are not in use, okay, they exist in two units, small subunit and large subunits. You already know that, right? But here's the thing. Small subunit is called 30S and large one is called 50S. But you put them together, now it is called 70S. What happened to the 10? Math. My math is bad. But this is correct. Yeah. Okay? That's your assignment for the next class, before the next class. Okay? Find out what happened to the 10. All right? I'll ask you that next time. And how about in eukaryotes? Remember, it's 80S a 50 and 40s. Small one is, no, this one is 50 and small one is 40s. No, not 30. In eukaryotes. In eukaryotes, this one is 40 and this one is 50. When you put them together, they become 80s. What happens to the 10? Okay? All right. So that's your assignment, to figure out what's going on. All right. All right. No sterile lipids in plasma membrane or cell membrane. What is the significance of this special lipid? Okay. What are 3 common things in all living cells? Okay, you can bet that that's definitely on your exam. 3 things that are...
Reproduction, self-reproduction.
Oh, no, there are 3 things that are found in a living cell. That are found in a living cell. All living cells, doesn't matter, prokaryote or eukaryote. All living cells have these 3 things. Okay? DNA, nuclear material, sure, good. Pardon me? Carbohydrates, yeah, that's too general. Okay, to make it broad. How about carbohydrates are found in the cytoplasm, protoplasm. Okay, good. So number 2, cytoplasm, protoplasm. Right? So DNA, cytoplasm, inside the cytoplasm. Yep. Cytoplasm. Good. Yeah. Now you have to confine all these things in something, right? Cell membrane. 3 things. 3 things. Cell membrane. Cell membrane. All living things, they have 3 things in common. Plasma membrane, cell membrane, cytoplasmic membrane, cytoplasm, and DNA. Okay, remember that, please.
All living things.
All living things. Now, their chemical composition may be different, but they all have these 3 things. All living things. Now, here I'm telling you, in prokaryotes, brain does not have sterile lipids. In sterile lipids, there are special type of lipids that are only found in the membranes of humans and other eukaryotes. Okay? DNA or nuclear material. Okay? Number 2, cytoplasm. And number 3, cytoplasmic membrane or cell membrane or plasma membrane. Okay? Now,
Or plasma membrane?
same thing, different names. Professor Khan, Mr. Khan, Master Khan, same thing. Same thing. Yeah, really? Same thing. All right. All right.
So I'm not sure what you said were sterile lipids.
I'm coming. I'm going to. A person sees a spot on their skin. They go to pharmacy and they pick up this ointment. Okay, and they start applying on it. Without knowing if this is bacterial infection or bacterial disease or fungal. Do you think it will work? Probably not. Okay. You need to know if this is bacterial or ringworm. Ringworm. What type of worm causes that? A ringworm infection. It's a fungal, it's not a worm. Okay, so ringworm is caused by fungus. Okay, so if the person sees this ringworm, okay, they go to the pharmacy and they say, oh, it's a kind of red rash, let me pick up some penicillin ointment. And they keep on applying. They can buy penicillin, but they go to the pharmacy, give me something for this. All right, it's not going to work. Okay. And ringworm, you have to apply for at least 3 to 6 months for that to disappear. Some people, they apply homemade remedy, bleach. Okay, no, don't do that, please, okay? So the medical significance is that if it is bacterial, okay, and you keep on applying antifungal, it's not going to work because bacteria don't have those sterile lipids. You must apply, if it is fungal, okay, like athlete's foot, You know athlete's foot, how long does it take to treat athlete's foot infection? Gone. Oh, that boom bomb it doesn't work. 3 months minimum, 3 to 6 months minimum. And you have to keep on applying. And then it may go away if you are a hygienic person. Otherwise, no, it takes too long. The person with fungal infection is that because they are eukaryotic. Okay, you cannot use high concentration of medication to kill fungus, unlike bacteria. Bacteria, they have so many differences. Fungi are like us. That's why it takes too long to treat fungal infection in smaller concentration for a very long period of time. Okay? Yeah. Nail infection, nail fungus, okay? How long? 3 to 6 months minimum. Yeah. Alrighty. Let's move on. What?
I don't understand. Sterile, I don't know.
Sterols are special lipids, okay, that are only found in eukaryotes, and their medical significance. They are, let's say, for example, let me give you a very specific example. A person has, they are found in fungal cell wall, and our cell walls, our membrane. Did I say cell wall? I've been keep saying cell walls.
Yeah.
Pardon me. Sorry. No, membrane. Okay.
Sterile
They're
lipids are found in fungal
cell membrane and human membrane, or eukaryotic membrane. Membrane. Now, if a person has bacterial infection, can you apply an antibacterial ointment to treat disease that is caused by bacteria? If you have a skin infection caused by bacteria, can you apply antibacterial to treat antifungal to that? Why would it not work?
You just switch from bacterial to fungal.
Yes.
No.
No. Because bacteria do not have sterols in their membrane. Fungi have. After class. Okay? All right. Similarity. What are some of the common things between prokaryotes and eukaryotes? Both prokaryotes and eukaryotes, we all contain 4 organic compounds. How many organic compounds do exist? How many? 4 or 5 or 6? Organic compounds, chemistry 1032? 4? Yes, there are only 4. Just double checking. All right. Nucleic acids. All living cells, with the exception of viruses. Viruses are exceptions. They never have DNA and RNA. All living cells, they have both DNA and RNA. Except viruses that have either DNA or RNA. Living cells always have DNA and RNA. Proteins. Why are proteins important? They give me the 3 most important functions of proteins in living cells. Number 1. Okay, they are essentials to make new cell components such as cell membrane. Okay, number 1. Okay, number 2. No enzymatic activity can take place without proteins because all enzymes are made up of proteins.
And transport of material based on
That's in the membrane. Yeah. All, number 1, new cells and cell components are made up of proteins. Number 2, all enzymes are made up of proteins. And number 3, all antibodies, not antibiotics, all antibodies are what? Proteins. All antibodies are proteins. And there are several other functions that we will talk about later. Okay? We both have carbohydrates. What do we do? What is the most important function of carbohydrates? Energy. Fuel. Lipids. What do we do with lipids? What do you think bacteria do with lipid? For the construction of cell membrane. both bacteria and humans, we carry out chemical functions to metabolize food. Did I explain the term heterotrophs and autotrophs to you? Heterotroph, okay. I think I should have asked you this, and I think I did, but maybe never answered this. Why all scientists who were conducting experiments in favor or against spontaneous generation, they use some kind of broth, right? Sometimes they use, like John Needham, he used broth, okay? Splanzani, he used broth, okay? Louis Pasteur, he used broth, right? Why couldn't they conduct the experiment with water, flask of water? Okay. This, yeah. Pardon me? Yeah, no food. Most microorganisms that we encounter every single day, they need to metabolize food. They need pre-synthesized food. Like if you sit down in front of TV with remote control in your hand, okay, for 2 weeks, do not stand up, do not eat or order pizza. Can you make your own food sitting in the couch? No. We are like most bacteria. Bacteria and human beings, we are similar. We need to, is it time? Oh, sorry. Please tell me 5 minutes in advance, okay? Yeah, please. Okay, thank you very much. Okay, here.